Hello, my dear students of class 12 science. Once again, welcome to environmental science class. We are dealing with the topic classification of technique for monitoring the ambient air sampling. Ambient means outdoor air sampling because the air contains different types of pollutants and which we are trying to analyze. So we have got different types of devices by which we can collect the air sample and we can analyze it. So some of the devices, they are easy to use and some of them are complicated. Okay, and the type of devices that we use or that we choose to use to collect the air sample and to monitor depends on what particular pollutants we are going to analyze that is present in the air, and in what particular place or what particular locations we have to use it. Okay, so depending on these particular situations, we have to choose particular devices that can be used to collect the ambient air sampling and that can be used to monitor it. So in the last class we have learned about the two groups or the two techniques which we can use to analyze the ambient air sampling. The first one we have learned was the manual technique. Okay, manual, which was user friendly, okay, which does not need electricity, and since it is easy to use, it is user friendly, it can be used in multiple locations. And under the manual technique we have learned about the high volume sampler, which is mainly used for collecting the sample of a particular matter present in the ambient air. Then we have learned about the bubbler technique. So these two particular technique, the high volume sampler and the bubbler, we have learned under the manual technique. Now today we will discuss about the next group or the next technique, which is called the instrumental technique. Okay, so what does instrumental technique means? Now when we are learning about manual technique, we learn that this manual technique, it was labor intensive, okay, and provide limited information, but they were cheap, they were easy to use, they do not need electricity, and it can be used in multiple locations since it was user friendly. But when we look at the instrumental technique, they are more sophisticated compared to manual technique. They are complicated. Okay, they are more expensive, but they are more efficient. A large number of data can be collected from this instrumental technique, but since huge amount of data will be collected, it will be difficult to analyze those data. Clear? So under the instrumental technique we are going to learn about the photometric technique, non-dispersive infrared technique, and the Chem Illuminations. Chem illumination means the property to emit light. Okay, so one by one we are going to discuss these three techniques under the instrumental one. Clear? So first of all we will learn about the photometric technique. Photo means what? Light. Okay, just the term photo means light. So in this photometric technique, okay, first of all the gas or the air sample means the gases, you can see over here. The gas to be evaluated is mixed with the reagents and passed through a photometric devices. So in the given figure you can see over here, this air sample means the gases which we are going to analyze, which we are going to monitor, because the gases which we are collecting as a sample contain different types of pollutants. We have already talked about air pollutions, right? So in that collected air sample we will get different types of pollutants, okay, which will pollute the air. So since those pollutants are present in the air, now in this particular technique we are going to find out the concentration of those pollutants, okay, which is present in the air. So those air sample which we have collected and which we are going to evaluate it over here will be mixed with a chemical or the reagents, you can see over here. So the air sample or the gases will be mixed with the reagents, and once they are mixed with the reagents, these particular gases will now pass through the photometric devices. So you can see over here, this is the photometric devices. So once the gases which are going to be analyzed is mixed with the reagents, is mixed with the chemicals, now they will pass through the photometric devices. Now the most important thing is that the pollutants which are present on the gases, now they will absorb infrared, they will absorb the ultraviolet rays, or they will absorb the radioactive rays. Clear? Once these gases will pass through these photometric devices, they will absorb this infrared, ultraviolet rays, or the visible radioactive rays, which can be measured. Clear? And those gases will absorb this particular energy. Energy means those infrared, ultraviolet rays, or the radiations at a particular wavelength. Clear? So what we are doing over here, we are mixing the gases with the reagents, and after mixing, we are passing these gases through the photometric devices. Now these photometric devices, or the photometry, uses the absorption of ultraviolet or infrared or the radiations by the gases, because the gases will absorb this energy at a particular wavelength. Clear? Now the loss of radiation intensity by these gases will be measured continuously, because since these gases is to absorb those energy, infrared, radiation rays, those ultraviolet rays, and once they will pass through the photometric devices through the photocells, they will start to lose the radiations, they will start to lose the energy, and this loss of radiation intensity can be measured continuously, and it can be used to analyze the pollutants present in the air. So by the help of this photometric technique, almost all the gaseous air pollutants can be measured, can be analyzed continuously. Okay, so photometer, it's mainly consists of radiation sources. You can see over here, the radiation sources means we is mainly giving the radioactive rays to the gases that is passing through the photometric devices. So over here, which particular part of the photometric devices will give the radioactive property to the gases passing through it. So in the given figure, you can see over here, the photometer is mainly consist of a radiation sources, this particular one. Okay, it has a focused beam of light. So it will continuously emit the light. It will continuously focus the light through a cell on a radiation detector. Clear? So this one will continuously emit the light that give the radioactive property to the gases passing through the photometric devices. And we have got the radioactive detector, which is called the radiation detector, and this particular detector will transform those beams of energy into the electrical signal. So those radioactive energy, those energy that like we are talking in terms of infrared or the radioactive or the ultraviolet energies, those energies will be transformed into the electrical signal. And once those particular energy will move through the photometric devices, the radiation detector of the photometric devices will evaluate, will analyze those particular energy. And since we have learned that when the gases will pass through the photometric devices, there will be loss in the intensity of energy or radiation. And this loss of radiation intensity can be used to analyze the presence of a particular pollutant in the gases. So therefore, once the gases will pass through the photometric devices, they will start to lose the radiation intensity. So now the loss of radiation intensity is a measure of its concentration. So whatever the type of pollutants will be present in the air, in the collected air sample, that can be analyzed, that can be monitored by the loss of intensity in terms of loss of radiation, in terms of loss of infrared or the ultraviolet energy by the gases. Clear? So by this way, by the help of the photometric technique, we can analyze the ambient air sampling. So what you're doing in this particular technique? In this technique, first of all, we are collecting the ambient air sample, we are mixing it with the reagent or the chemical, and we are passing it through the photometric devices. Okay, photometry, we use just the absorption of infrared or the ultraviolet or the visible radiations by the gases or by the pollutants. And when the gases will pass through those photometric devices, there will be loss of radiation intensity, which can be utilized, which can be used to analyze or monitor the types of pollutants present in the air. So by this particular technique, we can continuously monitor the type of pollutants present in the ambient air sample. Clear? So in this particular photometric devices, we have got the radiation sources. You can see the light in the figure. That particular radiation sources will give the property of radiation to the gases, to the air passing through the photometric devices. And we have got the radiation detector that will detect the radiation or the concentration or the intensity of radiation present in the air sample passing through it. So whenever the air will pass through the photometric devices, this radiation detector will detect the loss of radiation intensity in the gases and will continuously monitor and analyze the ambient air sample, which will give us the idea of the presence of particular pollutant in the air. Clear this one? Okay. Next one is the non-dispersive infrared technique to monitor the ambient air sample. Okay. Now these devices can also be used to monitor, to analyze those gases which used to emit the infrared radiation. Okay, remember one more time, this non-dispersive infrared technique is used to analyze those gases who used to emit infrared radiations, like gases like sulfur dioxide or the oxides of nitrogen, carbon dioxide and the hydrocarbons. Okay, and this particular technique has low sensitivity, so therefore it is far more better to use this technique to monitor the gases at the source emission, a particular source from where the gases are being emitted, rather than going for the collection of the ambient air sample, the outdoor air sample. This technique can be used to analyze the air at the sources from where they are being emitted. So it will be very, very helpful, okay, at the point sources from where these gases are being emitted, are being released into the atmosphere. And this particular technique is very commonly used for analyzing the air that contains carbon monoxides. Clear? Okay, now let's look at the figure in the second paragraph. Now the analyzer, which is mainly used to analyze the gases that emit infrared radiation, does not allow the dispersal of light. That means, like the prism which should split the light, the analyzer that is being used in case of NDIR does not split the light, but rather it will produce a band of light. Okay, so this analyzer that is used in NDIR is not splitting the light, but rather it is producing a broad band of light by the help of a band pass filter. Okay, and when these gases will pass in the form of a broadband light, then this analyzer, okay, will produce a signal. You can see over here, a signal will be produced. Clear? A signal will be produced over here once the gases will pass through this particular analyzer. Now what is do that the signal that is being produced, okay, the signal that is produced when the gas will pass through the analyzers will has to be coincided, will has to match with the absorption peak of the pollutant molecule. That means the peak of the gases which is being produced after being passed through the analyzer through the NDIR has to coincide, has to match with the absorption peak. This is called the absorption peak. Okay, so the absorption peak of the pollutant present in the gases has to be similar, has to be same with the absorption peak of the pollutant molecule. That means beforehand only we have to find out the absorption peak of the pollutant molecule, and if the same type of pollutant molecule is present on the gases, then those signals will definitely match with each other. So what we're doing over here, you can see over here the analyzer which is being used, it does not split the light. Okay, it does not split the light, but rather it will form a broad band of light. And the broad band of light is being produced because the analyzer uses a band pass filter. So when the gases will pass through the analyzer in the form of a broad band of light, and once it passes through the photo acceptance unit, it will form a absorption spectrum. Okay, it will form a absorption peak. Okay, so here this particular line indicates the absorption peak of the pollutant present in the gases that has passed through the analyzer of the NDIR. So the absorption peak of the pollutant present in the gases has to coincide with that of the absorption peak of the pollutant molecule. So if the absorption peak of the pollutant molecule and that of the gases that are part of the analyzer will coincide, will be similar, then we can say that particular type of pollutant is present in the gases. Let's say this is the absorption peak of our carbon monoxides, which we have already analyzed. Okay, now the gases that are passed through the analyzer of the NDIR also has got the same absorption peak. Then we can say that the carbon monoxide is present in the ambient air sample or the point source sample. So by this way we can analyze the presence of sulfur dioxide, carbon monoxides in the collected air sample. Third one, we have got Chem Illuminations. This particular technique is based on a reaction that produces light. It's very important. Okay, so Chem Illuminations is basically a property, is basically a reaction that produces light, because Chem Illuminations is the property of emitting light. So this particular technique is totally dependent on the technique of emitting light. Okay, now this technique is used to detect the oxides of nitrogen, which may present very small amount. Now Chem Illuminations is based analyzers, like they are not only used to analyze the particular pollutants present in the air, but they are also used for the combustion system also. Okay, so they are effective in analyzing the collected air samples, but the maintenance problems is there with this particular technique, because the analyzers that we are using in these techniques has to be maintained continuously. Okay, so we have got the problems with this particular technique regarding the maintenance of analyzer. Okay, but it is one of the most effective technique of monitoring the collected air sample. So let's check it out the principles behind this technique. Okay, so when nitric oxide's molecule react with ozone, it forms nitrogen dioxide in excited state. Let's look at the paragraph and explain. Okay, so a small fraction of these molecules, they will start to decay by emitting the photon, and when this photon, means the unit of light, it will go and react with the ozone, it will emit light. So the amount of light that is being produced, that is being emitted when the oxide of nitrogens or say start decaying by producing photon, and when the photon react with the ozone, what is being produced? Light is being produced. Okay, so the amount of light that is being emitted gives us the idea, gives us the measurement of the concentration of nitric oxide present in the sample. So we can say, in the given figure, we have got the ozone generator, we have got the air sample, and over here, whenever we learn that whenever nitric oxide molecules reacts with the ozone, it produces nitrogen dioxides. So therefore nitrogen dioxides will be produced. We have learned that when the oxides of nitrogen come in contact with the ozone. Now this particular oxide of nitrogen that is being produced after reaction with the ozone, they start decaying. But once they start decaying in the reaction chamber, they start producing what? They start producing light. They start emitting light. Now whatever the light will be emitted when the nitrogen dioxides will react, or with the nitric oxide will react with the ozone, then light will be emitted. So over here in this particular one, light will be emitted. So in this particular chamber we can say light will be emitted, and whatever the amount of light will be emitted over here by the reaction of the photon, by the reaction of the oxide of nitrogen with the ozone, those emitted light will be detected by the presence of photo detector. So depending on the concentration of the light emitted, okay, so the amount of light which is being emitted by the reaction of the photon, by the reaction of the photon with the ozone, this emitted light will give us the idea of the concentration of the pollutants like nitric oxide present in the air sample. So one more time, this Chem Illuminations is a technique which is based on the property of light emission, okay, and it is more effective for analyzing the ambient air sample, but that's mainly a problem with maintenance, okay. So here this technique is used to detect the presence of oxides of nitrogens which is present in very small amount in the air. Clear? So when nitric oxides present in the air react with the ozone, it forms nitrogen dioxide in the excited state. Now the small fraction of this molecule, that is nitrogen dioxide, which is being produced, but it starts decaying by emitting photon. Now that photon will mix with the ozone and produces light, will emit light, and that emitted light will be detected by the photo detector. So in the reaction chamber, the photon which is being produced after slowly deterioration of those nitrogen dioxides will produce the photon. The reaction chamber, the photon will react with the ozone and produces the light. That emitted light will be detected by the photo detector, and therefore finally the amount of light emitted will gives us the idea, will measure or will analyze the concentration of the pollutants like nitric oxide present in the ambient air sample or in the point source sample. So here we have learnt about the three techniques under the instrumental technique. The first one was photometric technique, the second one is NDIR, non-dispersive infrared technique, and the third one is Chem Illumination. So here we have finished the techniques which are being used for the collection of the ambient air sample, and by which we can analyze the air regarding the presence of different types of pollutants present in the air sample. Clear? So tomorrow we are going to have a live session, and there we'll discuss about this particular topic. If you have any confusions, we will discuss in that live classes and we will try to clear your doubt. Clear? So this much for today's class. See you in next class. Thank you.